Hello everyone, welcome back. So, in today's segment, we will be discussing a new unit of class 12. All right, so this is basically the unit 6, and it is based on the broader concepts of genetics. So, in this video, specifically, we will be studying the chapter 5, that is principle and of inheritance and variations. Just a moment. Yes. So, basically, uh, in this video particularly, we will be discussing what is genetics and how today we have come a long way and we have made many advances in that field. But to begin with, how did it start and how was it discovered and how did we name the term genes, or how do we know that what passed from one generation to another? And we will also learn about some concepts about given by the scientist Sir Gregor Mendel, and also how did he come to a conclusion about genetics, and on what basis did he make his law? What plant did he select to carry out his experiments? And we will also see an interesting concept called as test cross. So to begin with, I'm Janani Saptharishi, and I'm going to be teaching you that means class 12 biology. And what you'll get with Unacademy is that you will get daily live classes. All right. So you can choose any entrance exam you wish to crack, and for all the subjects there will be daily live classes along with live tests and quizzes. You can avail all the material you require, all the notes, all the lectures and everything. You have unlimited access to all the information. Also, we provide you structured courses. By that I mean that we assure a complete syllabus coverage. All right, so any exam you select, for example, if you're appearing for your NEET or preparing for your NEET, all you have to do is get the subscription and select your examination. And even if you join through my code, it is not that you will get content only of biology. You will be provided with your content for required for this entire examination, that is physics, chemistry, biology and so on. So you have everyday live classes with the educators that are top board with Unacademy. So with top educators, I mean that you see they have the blue ticks and you can study and optimize all your time with all of them. And it's up to you with all the ease and comfort. You may decide with which educator you're comfortable with and how you want to go on with the portion. So we have a very comprehensive syllabus. All you have to do is go to the Play Store and download the Unacademy Learning app and get its subscription. Once you have subscribed, all right, for more information or to talk to us directly, you can go to the Telegram app and copy the following URL. All this is given in the description box below. So when you get the subscription, these are the fee structure options you will be shown. Out of that, I recommend you most the 12-month one, because not only is it very economical, it is only 15,000 rupees for an entire year. It is not only very economical, but also to prepare for an examination like NEET, you'll at least require an year. So if you're in grade 11, I recommend you this highly. But if you're in class 12, you may go with any of the crash course mentioned. And another gift I would like to give from my side is that if you use the code Janani, that is my name, all right, you can avail and 10% offer on all the courses. So, so let us begin. Now, let us start with the introduction of the topic from the term what genetics means by itself. All right. Hi Nandini Verma. Hi Dia. नीट के लिए यूज होगा, हाँ ये पूरा लेक्चर नीट के लिए ही है, ठीक है. What is genetics? So genetics is basically a branch of biology that will deal with the mechanism of inheritance and variation. By inheritance, what do I mean is how a trait or a gene is passed from one generation to the another. We look into it in detail. And by variation, I mean that, see, we all know that all have studied the characteristics of the sexual reproduction, and out of this, variation was also termed as a genetic advantage. Why? And what causes variation? It is the recombination or the mutations that we see in every species. So we'll see that how the species are very much similar to the parent species, but they are also very much different, that is very much varied. So see, this is the hereditary we get from the parents. So here in the family, we can see that both the children resemble their mother. That is, as we say that like begets like. So what happens is similar characteristics are passed from one generation to the another, but they are not completely similar. So genetics basically will deal with the inheritance as well as the variations of characters from parent to offspring. So this is how. So we have like this example is shown that as we know that see, we have 46 chromosome in total. 23 we get from our mother and 23 from the father. So this is how we have shown that how there is a carrier father and a carrier mother. So basically carrier means it is they have a carrier, you can see, of some trait or even a disease. So from the progeny we can say either it see the yellow represents the normal part here. All right, so either it can be half and half, that is they give birth to a normal male, or they have a variation like this and this together will make a carrier female. Similarly, a carrier male can also be produced, but if both the affected genes flow through, they will give rise to a affected female. So we'll discuss many such examples in detail. So, however, see, as I told you, like begets like. That means I mean that all any species will give birth only to their species. So this is a line taken from the NCRT. That is, have you ever wondered why an elephant gives birth to only a baby elephant? Because of the flow of the genes, or in the broader terms, you can name is that name it as genetics. So this is a rare photograph showing DNA relation. All right, person in the left is the old grandfather, and this is his grandson. But we can see that they almost look similar to each other. This is somewhat a picture that will show DNA relation, or that means we can conclude that all the it is a good example of genetics or the hereditary flow. So yes. So the person in the left is grand old father and the right is the grand old son. So nearly more than 5 generations. So from all the generations, the genes have flowed from one generation to the another. So this is again some examples of siblings having a similar features, because siblings, as we all know, they will have the same parent genes. So both will have a similar genetic content, but not exactly similar. They will have some variation. Yes. So, the best examples for recognizing some gene traits in humans is clasping hands, tongue rolling. So, hands is basically when you put your clasp your hands together. If right is over left or left is over right. Tongue rolling we are all familiar with. Hitchhiker's thumb is basically if your thumb is exactly straight, or when you even in a relaxed position, it has a bend in it. All right. Earlobes attached or detached, and bent little finger is again same. So here we can see this is the example of clasping our hands. If the right is over left or left is over right. Then we have free or attached earlobes, followed by the hitchhiker's thumb. So see here, it is bent more than the normal position. That is a condition called as hitchhiker's thumb. So these are basically genetic characteristics. Means it if some people may have it this way, some people will not. So this is the rolling of the tongue. If you can roll your tongue, it will be somewhat like this. Fine. Yes, so this is another very good example of genetics, is that father and son taken photograph of the same age. So they look somewhat similar. So that is again an example for the hereditary. Now coming to identical twins. So identical twins are exactly similar looking. So we can say that we all bear some differences, but not the identical twins. They do not will bear much differences phenotypically, but genotypically. That means the genetic content, the features may be same, the external features, but the internal the genetic content will definitely vary in all organisms, even in identical twins. Fine. Genetics not only deals with inheritance, that is not only between the flow of the characters, but it will also deal with variations. So basically, it will deal with inheritance and variation. These are two very important concepts of genetics. So, what is meant by variation? So, variation is basically the degree by which the progeny will differ from their parents. As I see how varied it is. So, the offsprings, all right, how different they are from their parent. That is the variation cost. And what is the inheritance? Inheritance is simply the process by which the characters are passed from the parent to the progeny. Variations that were naturally present in the wild populations of the plants and the animals to selectively breed and choose the organisms possessed with desirable characters. So, what happens is that we humans also sometimes prefer artificial selection. That means, especially what we do in plants or the cattle are we artificially select the genes and make them breed so that we have a better quality. So that is what is mentioned here. So variations are naturally present in the wild population of the plants and animals. So we can select and breed and choose the organism that are possessed with the desirable characters or the better quality, or however is a requirement. So, Sahiwal cows in Punjab, these are all the genetically bred ones. That is, we have chosen the best genetic qualities and we have tried to artificially breed it. So, it is just an example. But now we will look into detail how Sir Gregor Mendel started with his experiments. Hello, Harman. So, this is Sir Gregor Mendel, and he is known as the father of genetics. All right. And he was the first to demonstrate the mechanism of transmission of the characters from one generation to the next generation. So, what he did was he basically studied कि parent generation से offsprings में characters या फिर कुछ traits कैसे flow हो रहे हैं. ठीक है. तो क्या हुआ कि उन्होंने डिस्कवर किया था समथिंग नोन एज बेसिक प्रिंसिपल्स ऑफ हेरिडिटरी. वी विल डिस्कस अबाउट दिस इन डिटेल. एंड उन्होंने कौन से ऑर्गेनिज्म में स्टडी किया था? इट इज द पी प्लांट और द पाइसस सैटिवियम और कॉमनली नोन एज द गार्डन पी. Clear? Yes. So he published the result of his studies under the name of Experiments on Plant Hybrids. Another very important point. This is the name of his journal, Experiments on Plant Hybrid. Yes, it was the pea experiments that we are talking about. So, for the first time, Mendel concluded hybridization experiments on garden peas for 7 years. All right, he studied the garden pea for 7 years, and he proposed the laws of inheritance. Again, very important. We have three laws in this. Fine. So, Mendel applied statistical analysis and mathematical logics to the problems in biology for the first time. And because he used statistical and mathematical methods, his work was not initially recognized. It was only when Sir Gregor Mendel died, his journal was, found, and his work was appreciated, and he was awarded the Nobel Prize after he died. So what happened is, since he, uh, started a combination of mathematics into biology, it was very new to people. And also in that times, people did not understand statistics and mathematics so well. So what happens is, and especially the problems in biology, it was introduced for the first time. So he did not gain much of acceptance. So what he did basically, as we have known as the garden pea, if you remember, we have studied previously that it is the mode of best example for self pollination, because they were clistogamous or closed flowers. So what he did was, he basically, see, he opened the flower and he tried to cross pollinate the flowers. All right. He tried to cross pollinate the flowers, and then he observed that if I cross pollinate the pea plant, is there any difference? Is there any difference in traits or anything like that? And if any doubts, you can put it up in the chat section. So, Mendel investigated several characters in the garden pea plant, and he also noticed that many characters are manifested as two opposite traits. So, what was the interesting thing about the pea plant? That all the trait it had, it basically was complete the opposite. It either had tall or dwarf, either completely yellow or green seeds. So there was nowhere in between this. All right. So it was either tall or dwarf, either yellow or green, in that way. So what he did basically was, he tried to cross between the tall and the dwarf plants. So he took the plants having two different traits and he cross pollinated or tried to cross them. And once he crossed a tall and a dwarf plant, he obtained some interesting results, or even you can cross a yellow and a green seed. That is just an example. So see, this is Mendel working with a pea plant in his garden. So this is how a pea flower looks, and so what he found was, see, what are this he first made a list of characters and its contrasting traits. As I showed you that whatever the two traits, they are exactly very opposite to each other. So either it for the character of the stem height, so either it is tall or dwarf. Coming to the flower color, either it is violet or white. Flower position is either axial or terminal. The pod shape, that means pod is basically all the small piece which you'll see inside the outer covering, is basically are the pods. So they are either inflated or constricted. The pod color is either green or yellow. The seed shape, again important, round or wrinkle, and the seed color is either yellow or green. Yes, traits are the characteristics. So here comes an example of stem height. So this is a tall plant and this is a dwarf plant. Dwarf, that means short. Flower color, so pea either bears violet flowers or it bears white flowers. Fine. Flower position, it is either axial inflorescence like this, in the axial terms, and either it is terminal or in the end. All right. Now the pod shape, as you can see, it is the inflated pod and it is the constricted pod. This is the pod color. These are the green and these are the yellow pods. Seed shape, seed color. Now, moving forward, what happened that Mendel conducted artificial or the cross pollination, as I told, and he used several true breeding lines. So, by true breeding lines, I mean that the plants that have undergone self pollination. All right. So, what I mean by true breeding lines is that that the plant that has undergone self pollination, and it will show the stable trait of inheritance and expression for several generations. तो जो प्लांट ने सेवरल जनरेशन या काफी जनरेशन तक सेल्फ पॉलिनेशन अंडरगो किया है एंड उसके पास एक स्टेबल ट्रेट है, तो अगर मैं एग्जांपल लूँ एक टॉल प्लांट का, और समझो कि वो टॉल प्लांट ने 4 to 5 times सेल्फ पॉलिनेट किया है, तो क्योंकि उसके पास सिर्फ टॉल ट्रेट है तो वो डॉर्फ प्लांट तो बना नहीं सकता, तो मतलब उसने 4 to 5 times टॉल प्लांट ही बनाया है। ऐसे प्लांट्स को हम सेलेक्ट करेंगे। हाय सुनील कुमार वर्मा। So basically, he selected 14 true breeding plants. Another important point. Alright, he selected 14 true breeding pea plant and varieties which were very similar except for one character. So that one character that was not same became the contrasting traits. Alright. So for example, all the characters were same, but one was the tall character and one was the dwarf. So this tall and dwarf became the contrasting traits. All right. Now, advantages of selecting a pea plant, or why did Mendel decide that he would start working on a pea plant? This is because pea is an annual plant, all right, with well-defined characters. See, please don't be under the impression that annual plant is something that will grow throughout the year. No, annual plant means that it in 1 year it will complete its life cycle. All right. By that I mean we have studied about the reproductive phases. That is first the birth, or the planting of the seed, then the growth or growth period, then we have the reproductive phase and the senescence phase. So basically this plant will undergo all these phases in 1 year itself. It will start flowering and fruiting in 1 year. That is why he selected the pea plant, and it can be grown easily and it can be crossed easily. It could easily be cross pollinated also. So pea plant is bisexual, since pea plant by the nature or originally it will undergo the self pollination. So for the self pollination what you need is bisexual flowers. That means the anther and the stamen and the anther, the stigma and anther should be in one flower itself. So basically, that is why he chose a bisexual flower containing both the male and the female parts. And it can also be self-fertilized conveniently. Since it is bisexual, self-fertilization तो बहुत easy होगा pea plant के लिए. And coming to a very important point, that it has a short life cycle and it will produce a large number of offsprings. पी की लाइफ साइकिल जो होती है वो काफी छोटी होती है, क्योंकि वो एक एनुअल प्लांट है, तो वो 1 ईयर के अंदर ही अंदर अपनी फ्रूटिंग भी दे देगी। और एक और एडवांटेज ये है कि ये लार्ज नंबर ऑफ ऑफस्प्रिंग्स प्रोड्यूस करती है। So just a quick few questions. Who is the father of genetics? It is, as we discussed about him, Sir Gregor Mendel. Mendel conducted his experiments on which plant? It is the pea plant or the garden pea. Who proposed the law of inheritance? It is again Sir Gregor Mendel. What is the contrasting trait for stem height? The most discussed example, the tall and the dwarf one. What are the advantages of selecting pea plant by Mendel for his experiment? Cross-pollination, long life cycle, unisexual flowers, or short life cycle and produce large number of offsprings. So, cross-pollination is also an answer, but the most precise answer is that it has short life cycle and it will also produce a large number of offsprings. Fine. Now coming to the inheritance of one gene, or I will be discussing something known as the monohybrid cross in detail. So this is how a pea plant was crossed. So what it was that from the parental generation, the pollen or the anther was taken and it was crossed between the stamen and the carpel. Fine. So, reasons of Mendel's success. Why was Mendel so successful in his experiments? He selected, first of all, because he selected pea plant. If he would have selected some other plants, he would have either, he was lucky enough to get his hands on pea plants because some other plants, they could have some unusual traits or the traits which could lead into mixing up of the two characters and we could even see some um different traits like incomplete dominance and all, and which we'll study later. So he was lucky enough to get his hands on pea plant, and pea plant produces a large number of offspring. So basically larger the offspring, अपने probability तो पढ़ी होगी, तो सैम्पलिंग साइज जितना बड़ा है उतने हमारे लिए अच्छा है। तो क्योंकि लार्ज नंबर ऑफ ऑफस्प्रिंग्स है तो हमें ज्यादा सैम्पलिंग साइज मिलता है, तो उससे क्या होता है कि उसके डेटा ज्यादा हम एश्योर कर सकते हैं कि ये जो डेटा है वो सही है, तो इट गिव्स लार्जर क्रेडिबिलिटी फॉर द डेटा. And he started hybridization experiments considering single character at a time. So basically हमने जितने भी characters देखे, हमने 7 देखे थे, तो उसमें से उन्होंने हर एक कैरेक्टर को एक एक सिंगल कैरेक्टर को स्टडी किया. And then statistical and mathematical logic were applied. So when he bought new terms like the Punnett square and the dihybrid monohybrid cross and how we can calculate the ratios, these all things were very essential, and it also basically proved, and it you could not deny those mathematical facts. So that was his reason for success. So inheritance of a single gene or a monohybrid cross. What is meant by a monohybrid cross? Let me discuss that. So basically a monohybrid cross is a cross between two parents. Alright, so between two parents, but these parents will have a different character but a single different character. मतलब कोई एक character ऐसी होगी जो इन दोनों parents में different है. तो जब हम किसी ऐसे पेरेंट्स को क्रॉस करते हैं तो उससे कहते हैं मोनोहाइब्रिड क्रॉस, जब केवल एक सिंगल कैरेक्टर डिफरेंट हो. अगर हमारे पास दो कैरेक्टर डिफरेंट होता तो उसे हम कहते हैं डाइहाइब्रिड क्रॉस. फाइन. Yes. So Mendel crossed all the tall dwarf pea plants to study the inheritance of one gene, and he collected these seeds and produced as a result of the cross and grew them to generate plants of the first hybrid generation. तो बेसिकली जो भी हमें क्रॉस करके जो पहला रिजल्ट मिलता है उसे हम कहते हैं फर्स्ट हाइब्रिड जनरेशन. So this is also called the first or the filial. Filial का मतलब होता है पहला यानी कि first, और इसे हम F1 generation भी लिखते हैं. Fine. तो ये देखिए, ये है हमारा पेरेंट जनरेशन, एक है टॉल एंड एक हमने लिया ड्वार्फ. ठीक है, तो अभी जब हमने इन दोनों को क्रॉस किया तो हमें मिला ये. तो इसे तो ये हमारा है फर्स्ट जनरेशन, तो इसे हम कहते हैं F1। ऑल राइट, एंड अभी इसका अगर हम टेस्ट क्रॉस करेंगे तो ये कुछ ऐसे आएगा। सी F1 gametes are listed here, ऑल राइट, एंड मतलब इसी को हमने इसके साथ क्रॉस किया टू गेट द F2 जनरेशन। तो T कैपिटल T स्मॉल t इंटू कैपिटल T स्मॉल t, so then we got a ratio of 1:3. Now another interesting thing we found out here was that चाहे कोई भी कैरेक्टर लो, जो फर्स्ट जनरेशन होता है वो एग्जैक्टली सेम ट्रेट कैरी करता है अपने पेरेंट का। जैसे यहाँ पे हमने अपने डोमिनेंट पेरेंट का, आई मीन, ऑलराइट, तो यहाँ पे जैसे जो कैपिटल टी है ये है डोमिनेंट, ठीक है, और ये है रिसेसिव, ठीक है। तो अभी जैसे हमने देखा ये टॉल जो डोमिनेंट है, फर्स्ट जनरेशन भी टॉल आया। किसी भी वे में फर्स्ट जनरेशन जो है वो रेसेसिव ट्रेट नहीं कैरी कर पाता, ठीक है। हम कभी ये नहीं देखेंगे कि टॉल एंड ड्वार्फ के बीच में क्रॉस हुआ एंड जो फर्स्ट जनरेशन है वो रिसेसिव यानी कि शॉर्ट आई। ये तभी पॉसिबल है जब भी टी 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 के बीच में क्रॉस होगा। I hope this is clear. See, so the first generation here is a capital T and a small t. And in such a condition, always the dominant character, that is the capital T, will dominate. That is, the plant is phenotypically tall. That means by the external, by my eyes, I can see that the plant is tall. Yes, I think Sunil Kumar Verma has answered your question. In monohybrid, we'll consider only one trait. So he made similar observations for the other pairs and traits and found that the F1 always resembled the dominant parent. So Mendel observed that the F1 progeny, like the plants, were tall like that one of its parents, and none of them were dwarf, and that the trait of the other parent was not seen in them. तो वही हम कह रहे हैं कि F1 में जो रिसेसिव पेरेंट है, मतलब अदर पेरेंट, उनका ट्रेट हम उसमें नहीं देख पाते। एंड मेंडल देन सेल्फ पॉलिनेटेड द F1 प्लांट्स। अभी जब इस F1 प्लांट्स को हमने सेल्फ पॉलिनेट किया, लेट मी शो यू, सी हियर, सो हियर वी हैव बेसिकली सेल्फ पॉलिनेटेड द F1 F1 traits. So when we self-pollinate, then we will see a dwarf plant and it will be in the ratio of 1:3. Why so? Because it is two capital T tall, a capital T and a small t. So the capital T will dominate, again tall, and similarly tall. So this is the phenotypic ratio. So, but if we see the genotypic ratio, that is according to the exact genes, how it is that it is 1:2:1. So, this is the genotypic ratio. I hope this is clear. So, Mendel, yes, and when he self-pollinated the F1 generation, what we saw, that is the Punnett square, was the F2 or the second filial generation. And from that, some of the offsprings produced were dwarf. So this dwarf character was not seen in the F1 generation, but only expressed in the F2 generation. So there are many objective questions that are asked. Yes, so there are many objective questions that are asked from these concepts. So you need to remember these. Now, the proportion or the probability of plants of the F2 generation is 1/4 are dwarfs and 3/4 are tall. So we have a ratio of 1:3. The tall and the dwarf traits were identical to their parent types and they did not show any blending. By that I mean, जैसे कि मैंने कहा था कि पी प्लांट में क्या होता है कि वो एग्जैक्टली ऑपोजिट ट्रेट शो करता है, मतलब यानी कि कुछ हुए या एकदम शॉर्ट, कुछ हुए एकदम टॉल, ठीक है। हमें कोई भी मीडियम लेंथ या इन दोनों के बीच का लेंथ हमें नहीं ऑब्टेन हुआ। That means that all the offspring were either tall or either short. There was nothing in between. So, what happens is only one of the parent trait was expressed in the F1. ये तो हम समझ गए अभी कि सिर्फ एक trait express हुआ F1 में, वो है dominant. And F2 में both the traits were expressed in a proportion, that is 1:3 or 3:1. Now, तो अभी हमने क्या देखा कि ये जो यूनिट है ऑफ इनहेरिटेंस, मतलब जो यूनिट है जो एक जनरेशन से दूसरे जनरेशन में फ्लो हो रहा है, वो है हमारे जीन्स. ठीक है. तो मेंडल प्रपोज दैट समथिंग वाज बीइंग स्टेबली पास्ड डाउन unchanged from parent to the offspring through the gametes over the successive generation. This definition is very important, taken as it is from the NCRT, and also some questions in NEET are based on this, directly on this definition itself. So इसे आप याद कर लीजिए अच्छे से. तो ये जो पास होता है फ्रॉम वन जनरेशन टू अनदर एंड इट इज अनचेंज्ड, these are called as factors. All right. And earlier it was called as factors, but later they were given a term known as gene. So genes are the units of inheritance. All right. Fine. They contain the information that is required to express a particular trait in an organism. तो क्या होता है कि जीन के पास वो सारे नेसेसरी इंफॉर्मेशन होते हैं जो एक ऑर्गेनिज्म को एक ट्रेट देता है, यानी कि एक ऑर्गेनिज्म को या तो टॉल बनाता है या ड्रॉप बनाता है, या उसे एक पर्टिकुलर कह सकते हैं फ्लावर को कलर देता है, वो पर्पल होता है या वाइट होता है, ऐसे. ठीक है. तो gene is the unit of hereditary, and genetics is basically the study of both hereditary and variation. So in those slides में मैं इस चैप्टर के सारे important definitions को short में represent करने वाली हूँ. आपको जो सही नहीं लगता है, yes, I mean जिसमें आपको डाउट है वो आप पूछ सकते हैं, और बाकी चीजें आप नोट डाउन करिए, एंड कुछ डेफिनेशंस हम बाद में आगे वापस डिस्कस करेंगे. So hereditary is transmission of characters from one generation to the another, that is parents to the offspring. Now coming to factor. So the trait determining thing, मतलब जो हमें ट्रेट डिटरमाइन करता है कि या तो वो टॉल है या वो शॉर्ट है या जैसा कलर है, वो सारे होते हैं फैक्टर्स, जिन्हें बाद में कहा गया था जीन्स. एलियल्स होते हैं ऑल्टरनेटिव फॉर्म ऑफ जीन्स, मतलब उसी फैक्टर में देखिए एक है टॉल एक है शॉर्ट, तो ये है ऑल्टरनेटिव, मतलब दो डिफरेंट टाइप हुए, तो इसे कहते हैं हम एलियल्स. अब आता है फिनोटाइप. फिनोटाइप मतलब मॉर्फोलॉजी एंड एक्सटर्नल अपीयरेंस, या तो हम उसे कह सकते हैं फिजिकल अपीयरेंस ऑफ अ कैरेक्टर, मतलब हमें जिस तरह से वो दिखाई देता है. Now, genotype is the genetic constitution of a character जो हमें दिखाई नहीं देता. Then coming to a term known as homozygous. Homozygous means a pair of similar alleles. मतलब या तो TT मतलब दोनों ही टॉल है. ठीक है. ये हो गया हेट्रोजाइगस, यानी कि डिफरेंट अलील्स, मतलब दो डिफरेंट ट्रेट्स का ये मिक्सचर है. ठीक है. यहाँ पे इट इज नॉट लाइक दैट, इट इज इधर completely dominant or completely recessive. So a pair of dissimilar gametes, that is the capital T and the small t, is heterozygous. Now coming to an interesting concept called as back cross. ठीक है. बैक क्रॉस में हम क्या करते हैं कि जनरेशन से पेरेंट का हम पेरेंट का हम जो भी, या वो होमोजाइगस है या हेट्रोजाइगस है, उसका हम ट्रेट फाइंड आउट करने की कोशिश करते हैं. दैट इज F1 बिटवीन एनी ऑफ द टू पेरेंट्स, and test cross is crossing F1 with the recessive parents. All right. Now, what is a dominant character? The character which is expressed in the F1 generation is always termed as the dominant character. I hope this is clear. Now, what is a monohybrid cross? So, monohybrid cross is basically, as I told you, it is a cross between two parents that are genetically different with a character. All right. So, a cross between a tall and a dwarf character. In the dihybrid cross, we can include two different characters. All right. So here, what we can see is we have capital T, that is small r. R, I think, is for the if I'm not wrong, it's for the round and the shape of the pod. So basically, with the capital T, capital R here along with the small r. Fine. So the dominant character, that as we saw expressed in the F1, the recessive which is not expressed in the F1, but it is expressed in the F2 generation. Now, what is termed as the locus or the position of the gene on the chromosome? So basically, if consider this as your chromosome and see here, there at a point where you see the color red and blue is basically the gene locus. Basically, it will tell you the position. All right. पोजीशन ऑन what? पोजीशन ऑन अ क्रोमोसोम. एंड ये किस चीज का पोजीशन बताएगा? ये पोजीशन बताएगा किसी भी एक ट्रेट यानी कि किसी भी एक एलील का. ठीक है, तो ये है एक होमोलॉगस क्रोमोसोम. होमोलॉगस क्यों? क्योंकि इसका जो लोकस है सेम जगह पर है. अगर ये इसके जगह पे ये यहाँ होता तो ये बन जाता हेट्रोजाइगस. ठीक है, क्योंकि ये सेम लोकस यानी कि सेम पोजीशन पे है बोथ द क्रोमोसोम्स में, इसे हम कहते हैं होमोलॉगस. What are alleles? So basically, genes with which code for a pair of contrasting traits are called as alleles. They are slightly different from the same gene, and we use alphabetical symbols for each gene. Then a capital letter is used for a trait expressed in the F1. तो जो भी F1 में आया वो हुआ dominant and उसके लिए हमेशा capital letter use होता है. And जो F2 में express होता है यानी कि recessive, उसके लिए हम small alphabet use करते हैं. For example, in the case of height, we can see T is the capital T is for the tall and the small t is for the dwarf one. So capital T and the small t are the alleles for each other. So hence, it is in plants it is possible that there are pairs of alleles. All right. तो जब भी ये जो allele का pair बन जाता है वो इन 3 combination में हो सकता है. तो अब मेंडल ने ये भी प्रपोज किया है कि जो ट्रू ब्रीडिंग यानी कि सेल्फ पॉलिनेशन जिनमें हुआ है जनरेशन्स में, वो टॉल और डॉर्फ की वैरायटी, ठीक है, इन्होंने लिया एंड द एलिलिक पेयर ऑफ जीन्स फॉर द हाइट आर आइडेंटिकल और होमोजाइगस. ठीक है, तो ये तो है कि जो भी ट्रू ब्रीडिंग है उनमें तो सेल्फ पॉलिनेशन ही हुआ है तो वो तो आइडेंटिकल ही रहेंगे. ठीक है, तो ये हुआ कैपिटल टी एंड स्मॉल टी जो भी हो. नाउ जीन इज बेसिकली डिटरमिनेंट ऑफ द कैरेक्टर्स प्रेजेंट इन द क्रोमोसोम्स. मतलब जीन्स उसे कहते हैं जो कैरेक्टर्स को डिटरमाइन करते हैं. ठीक है, एंड नाउ it is also known as the fundamental physical unit of hereditary because genes are transferred from one generation to the another. So he referred to them as the factors. Now allele or allelomorph, again coming to this point. So two Mendelian factors which will occupy the same locus on a homologous chromosome and that control the expression of the character. Very important line. Many questions are asked with this. Again, I'll read it for you. Two Mendelian factors or genes which occupy the same locus on the homologous chromosome and that which will control the expression of the character. So, for example, the gene of the purple color flowers and the white color flowers are located in the same locus of the homologous chromosome. See, here the position in the chromosome in both the traits are same, and they and hence This is the exact meaning of the word allele or allelomorph. As we have discussed, locus refers to the position of the gene. So allelic pairs on the homologous chromosome have their respective positions or the respective locus. Yes. Homologous chromosome. See, here is an example of homologous chromosome. So basically, they are morphologically and genetically very similar to each other. Fine. What would be the phenotype of a plant that had a genotype T and T? I hope this concept is clear. As I told you always, the dominant trait is expressed, so the answer will be a tall plant. So the capital T and the small t, or the tall and the dwarf, are called as genotypes of the plant, whereas while descriptive terms tall and dwarf are the phenotypes. All right. So basically, when we write it in such terms, these are referred to as genotypes. All right. And the term tall and dwarf represent its physical characters. That is why it is called the phenotypes. So Mendel found that the phenotype of the F1, that is the heterozygote, is exactly like the tall plant in that of the experience. So he proposed that a pair of dissimilar factors, that is, one dominates the other factor, and hence the dominant factor was one that was expressed while the other was the recessive factor. This is known as this is the first law, that is the law of dominance. If one character is not expressed in the F1, this also you should be able to answer. It is the recessive factor. So yes, these are some examples of the dominant and the recessive traits. Fine. Now, what happens in this case? The tallness is dominant or the dwarfness. And alleles can be similar in the case of homozygotes, and it can be dissimilar in the case of heterozygotes. And as genes are controlling one character, that is the height, if we see, the capital T and the small t, that is the heterozygote, is the monohybrid. All right. And the cross between this will be termed as a monohybrid cross. So again, the same thing. See, he observed the recessive parental trait, and it is expressed without any blending in the F2 generation. As we saw that we could not see any in between tall and dwarf characteristics. So they basically produce gametes by the process of meiosis. All right, very important and a probable question. So the alleles of the parent plant segregate from each other, and only one allele is transmitted to a gamete. So the segregation of alleles is a random process. All right. Now here we are studying about the law of the segregation. So there is a 50% chance or a say 1 by 2 probability of a gamete containing either of the allele. Alright, so since there is no mixing, it will either be tall, either be dwarf. So the probability is 1 by 2. The gametes of the tall plants have the capital T allele, and the gamete of the dwarf have the small t alleles. Now, during the fertilization of these two alleles, what happens is that from one parent through the pollen, the from the tall plant, all right, the pollen to the dwarf one, all right, from the other parent and through the egg are united to produce the zygote. So the zygote has one tall allele and one dwarf allele. Fine. So these hybrids are the capital T and the small t. So since these hybrid contain both the type of allele and they express something called as the contrasting traits, and hence they are termed as heterozygous. So this is an example of heterozygous condition. See, you can see here this, and just have a look here. Again here and here. Fine. So this is again some examples. Just a moment, I will just this. Yes. So this is again some example of the heterozygous gene pair. So what happens is that these polled cow also carry two different genes. Now, what is a Punnett square, commonly known as the checkerboard? As we know that Mendel used many mathematical and statistical concepts, so out of these, one is that these type of gametes are produced by the parents and the formation of the zygotes and the progeny are represented by a diagram. And this is commonly known as the Punnett square board. And it was developed by a British geneticist known as Reginald C. Punnett. It is a graphical representation to indicate all the possible unions of the gametes. So what is it is used for? इसे हम यूज करते हैं सारे पॉसिबल आउटकम्स देखने के लिए किसके यूनियन ऑफ गैमेट्स के कब देखते हैं फर्टिलाइजेशन प्रोसेस के वक्त इसे देखा जाता है. So the Punnett square method. So the Punnett square method, see, it is basically this. So here it is a cross between two heterozygous conditions. So we saw that the ratio phenotypic is 3:1 and the genotypic ratio is 1:2:1. So these three are the tall phenotypically and this one will be phenotypically short. Now, probability in relation to genetics. So, Mendel's law of segregation and the independent assortment reflect the same laws of the probability itself. So, basically, let's take an example of tossing of coins or rolling of dice. So, what happens that these probability are basically scaled from zero, that an event with no chance of occurring to an event of a certain chance of to occur. तो इधर नो चांस होता है या तो चांस होता है. तो जब नो चांस होता है प्रोबेबिलिटी में हम उसे टर्म करते हैं जीरो एंड जब उसे चांस होता है तो उसे हम टर्म करते हैं एज वन. So this is an example of the probability of tossing the heads. It is 1/2. Yes. So the parental and the dwarf produce gametes in the F1 progeny is capital T and small t. And the F1 plants of the genotype T and T are self-pollinated with each other to get the F2 generation. So I hope you know the symbols for the male and the female. So this basically is for the female and this is used for the male. So the F1 plant of the genotype, alright, this is the genotype of the F1 plant and they produce a gamete of the genotype capital T and small t in equal proportion. From that it can be seen that तो जो Punnett square से हमें results मिलते हैं वो है 1/4 यानी कि 25% of random fertilization that will lead to the completely dominant trait and half or the 50% will have this heterozygous condition and the last is that another 25% or 1/4 leads to the probability of the recessive state. Fine. So, 3/4 of the plants are tall and 1/4 is dwarf. So, externally, we can't distinguish between these two genotypes, all right? But, so hence the genotypes PT and TT are only tall is expressed in them. Hence, the character tall or the allele is said to dominate over the other character. Fine. So, due to this dominance, one character over the other in that of F1 are tall in shape. Now, yes, so this is the genotypic ratio that we observe. So the genotypic ratio, the probability is 25% or 0.25 to 0.550% to again 25%. So see, this is the mathematical genotypic ratio, and we can express this as a binomial expression. So when I put this into a binomial expression, how will it be? It would look something like this. Let's say 1 by 2 of the tall plus 1 by 2 of the dwarf. So this is my binomial expression. So when I start to expand it, I will first get 1 by 4 t square plus 2ab, that is 1 by 2 into 1 by 2 into 1 by 2 uh, t and t. So this will get cancelled, and again I will have 1 by 4 t square. So this can be written in such a form, that is 1 by 4, that is this capital T and capital T, 1 by 2 of the capital T and the small t, and 1 by 4 again of the small t and small t. So from this we get the probability relation or the ratio. I hope this is clear. So Mendel self-pollinated the F2 generation now, alright, and then he found the dwarf plants of the F2 plants continued to generate the dwarf plant in F3 and F4 generations also. So when the F2 generation was self-pollinated, हमें dwarf plant आगे के generations यानी third and fourth generation में भी दिखाई दिए. So इससे conclude हुआ कि progeny यानी कि जो जीनोटाइप ऑफ द ड्रॉप्स है उसका था होमोजाइगस कंडीशन. ठीक है. What is a monohybrid cross? You should be able to answer this. That is the cross between two parents in a single character. First generation of the monohybrid crosses, first generation is the F1 or the first filial generation. Mendel self-pollinated the tall F1 plants from F1, F2, he self-pollinated it from F2 to get F3 and F4. What are the unit of inheritance? It is the genes that are the unit of inheritance. So the alternative form of genes is basically termed as the alleles. Now, coming to the last concept of today, the back cross and the test cross. So basically, what is a test cross? By the word itself, test. Basically, हमें test करना है कि हम test करेंगे उसको एक recessive parent के साथ. Please remember the word recessive parent. So a test between the recessive parent to find out whether it is in this. These are the three probabilities which we can have. So when the genotype is unknown, basically we will cross it with the recessive parent to know the genotype. Clear? So in a typical test cross, an organism showing a dominant genotype, alright, is crossed with one having a recessive parent instead of self-fertilization. So, the progenies of such cross can be easily identified to predict the genotype. For what the test is done, it is basically to predict the genotype of the organism. Very important point. So, this results, alright, of a typical cross where the violet color flower, that is the capital W, is dominant over the white color flower. If the F1 hybrid is crossed with the parental type having the dominant trait in the homozygous condition, no recessive individuals are obtained in the progeny. This we know, alright? तो अगर हमने F1 जनरेशन को हमारे डोमिनेंट ट्रेट से क्रॉस किया, तो जो सारे जनरेशन होंगे वो सभी के सब टॉल होंगे, ठीक है. So although the ratio of the plant showing phenotypically dominant characters, they show that the genotypic ratio is 1:1. Fine. यहाँ पे जीनोटिपिक रेशियो 1:1 क्यों हुआ? क्योंकि क्योंकि क्या हुआ कि देखिए ये हुआ, इसके हुए 2 and ये हुआ 2. Alright, so 2:2, that is 1:1. Fine. Now, taking another example, this I am crossing the homozygous dominant with the homozygous recessive. Alright, always it will be crossed with recessive. So this is completely dominant genotype. So here all the uh, or offsprings that are obtained are basically the dominating one. It will have the character of the dominating one, that is, it will be purple. All right, so all offsprings are purple. Therefore, the unknown flower was homozygous. So, see, if I give you a purple flower, all right, yes, and I ask you if what are the alternatives. You can again, and if I tell you that all the, now how I'm telling you, explaining that how this test cross will work. So if I give you a purple flower, all right, sorry, purple flower, and I tell you that all its offsprings are purple in color, and you have to find out its parent genotype. All right, so what you'll do is simply cross it with the, just take both see, पेरेंट में या तो कैपिटल की ये होगा या तो ये होगा. तो नाउ यू हैव टू ट्राई बोथ एंड क्रॉस इट विद द रिसेसिव वंस. तो इन दोनों क्रॉस में से जिसमें भी आपको ऐसा कॉम्बिनेशन मिल रहा है जिसमें आपको रेशियो 4:0 मिलेगा, वो होगा उसका पेरेंट इस केस में. बट अगर मैं आपको बता दूं कि जो ऑफस्प्रिंग्स है उसमें हाफ ऑफस्प्रिंग्स वाइट है, हाफ पर्पल है, तो इसमें जब आप टेस्ट क्रॉस करेंगे, मतलब रिसेसिव के साथ क्रॉस करेंगे, तो जिसमें भी ऐसा कंडीशन होगा तो हम डिटरमाइन कर सकते हैं कि ये जो पेरेंट है उसका हेट्रोजाइगस है. ठीक है, तो हेट्रोजाइगस के साथ क्या होता है, हमें मिलता है 1:1 का रेशियो. Clear? So such questions are asked when the offsprings are in the ratio of 1:1. What are is the parent heterozygous or homozygous? Or if the parent is if the ratio is 4:0, how is the parent type? So here, how do we determine it? We determine it by a test cross. So test cross is basically a kind of the back cross, because from the offsprings we are tracing its parents. And what is a back cross? Back cross is basically when the F1 individuals or the heterozygotes are crossed with any of the one parents or individuals that are phenotypically and genotypically similar to their parents, and hence it is called the back cross. And test cross is one of the kind of the back cross. And Mendel proposed two general rules to consolidate this understanding of inheritance in monohybrid. Let us see. So the first is the law, principles of laws. The first law is the law of dominance. The second is the law of segregation. We have seen both. The what will appear in the F1 generation is the dominant, that is the this, and we have also seen that they undergo random segregation of these traits, and that is the law of segregation. Test cross means crossing the F1 with dominant parent, recessive parent, both the dominant and the recessive, or none. So test cross is basically always and always carried out with the recessive parent. Test cross is a type of monohybrid, dihybrid, back cross. Test cross हम dihybrid का भी कर सकते हैं. This we will study when we are studying in detail about the dihybrid crosses. And test cross is a type of back crosses. What is Mendel's second law? Mendel's second law is the law of segregation. Back cross means crossing F1 with dominant recessive any of its parent or F1. So basically टेस्ट क्रॉस होता है उसे क्रॉस करना रिसेसिव के साथ एंड बैक क्रॉस इज क्रॉसिंग विद एनी वन ऑफ इट पेरेंट्स. So we will start with the law of dominance and segregation in the next lecture. We will discuss about all the three laws, and I will also make you familiar with what are dihybrid crosses. So that's it for today. We will meet tomorrow at 11:30 a.m. at the same time. Please do like, share, and subscribe the channel, and please forward the link and do like the video. It is like a motivation for me. If you learn something new, please do like the video. Thank you.